Hello and welcome to the 38th Great Lakes Phragmites Collaborative webinar, part of our Applied Techniques webinar series. Our presenter today is Sarah Marshall, who will be discussing experimental controls for Phragmites at Rare Charitable Research Reserve. My name is Samantha Tank, and I'm at the Great Lakes Commission and hosting this webinar on behalf of the Great Lakes Phragmites Collaborative. Before we get started on our featured presentation, there's just a few housekeeping things that I'd like to review with you. The Great Lakes Phragmites Collaborative is a partnership to link people, information, and action on invasive Phragmites. The collaborative is coordinated jointly by the Great Lakes Commission and the USGS Great Lakes Science Center, with funding from the Great Lakes Restoration Initiative. The collaborative facilitates communication among stakeholders across the region, assists in the development of management tools and programs, and serves as a resource center for information on Phragmites biology, management, and research. The collaborative is led by a multi-agency advisory committee and applies principles of collective impact to advance a common agenda for Phragmites management in the Great Lakes region. Another way the collaborative engages with Phragmites professionals is through the Phragmites Adaptive Management Framework, or PAMF. PAMF is a program that utilizes participatory science to provide data-driven Phragmites management guidance via an adaptive management framework. What this means is that we've really established a way to learn from what Phragmites managers are doing, and in turn that data is turned into annual guidance that helps those managers make effective and efficient management decisions. We encourage anyone in the Phragmites community who is managing Phragmites or is considering managing and is within the Great Lakes Basin to join PAMF. We're planning to host training sessions throughout the Great Lakes region in the spring and early summer to help anyone interested get involved. For information on where those training sessions are being hosted, you can go ahead and visit the website on the page, or email me at pamf@glc.org. If you're interested in learning more, you can check out the other resources on our website, which include management guidance and recordings of past webinars. You can also join our listserv to connect with hundreds of other professionals and sign up for our bi-weekly newsletter, which includes a quarterly research roundup of academic papers published in the previous month. You can find links to join the newsletter and listserv on the front page of our website, and that's www.greatlakesphragmites.net. Today's webinar is going to be recorded and uploaded to our YouTube channel and website. You can access an archive of past presentations on our website. You can also view and register for upcoming webinar presentations by following the URL listed here. So hello to all of you who are listening to the webinar recording. You can please go ahead and share this with your colleagues, and don't hesitate to reach out even if you're watching this long after the presentation date. And of course, go ahead and connect with us on social media. You can tag us on your Facebook and Twitter posts as @GLFrag. To help the webinar run smoothly today, I just want to go over a few technical reminders. Audio for this webinar can be streamed through your computer speakers or your telephone. To use the phone line, go ahead and select the phone call option from your GoToMeeting dashboard and access the number to call into, as well as the conference ID number and the audio pin to connect to the webinar. And if you would like to use the computer audio instead, you can do that by going ahead and selecting that option. If you're experiencing any technical difficulties, go ahead and send a message to my colleague, Patrick, and you can do that through the chat box at the bottom of the GoToMeeting dashboard. You can select, it will say to, and right now it's selected automatically to everyone. You can go scroll down to Patrick Kenneth, and he's going to be monitoring that attentively. In order to minimize any potential for interruptions and background noise, we have automatically muted all attendees, and attendees are going to remain muted throughout the webinar. Questions can be submitted anytime during the webinar. Our speaker is going to answer all questions at the end. We're going to do our best to take questions or comments during the webinar after the presenter has finished. If you need to leave, however, before the webinar before we have gotten to your questions, we just encourage you to review the recording to hear your question and answers at that time. Now, it's my great pleasure to introduce our presenter. Sarah Marshall is a conservation technician at Rare Charitable Research Reserve. After graduating with an environmental biology degree from the University of Saskatchewan, Sarah moved to Ontario to pursue a career focusing on habitat management. She She received a graduate diploma in ecosystem restoration from Niagara College and then worked for 2 years as a habitat technician, reptile monitoring technician, and fieldhouse manager in the Simcoe, Muskoka area. She joined RARE's land management department as a conservation technician in 2019 and is responsible for executing land use plans for the property, such as invasive species, invasive plant management, trail maintenance, and plant restoration. So thanks so much, Sarah, for sharing your work with us today. I'm going to go ahead and transfer controls over to you.
Okay, hello.
Hi, Sarah. We can hear you loud and clear.
Sharing my screen. And okay, we're good to go?
Yep, looks great. Take it away.
Okay, so thank you, Sam, for that very kind intro. And a big thanks to the Great Lakes Phragmites Collaborative for inviting me to come and speak on this webinar series. As Sam said, I'm the conservation technician working at Rare Charitable Research Reserve. And I'm going to be talking a little bit about the Phragmites research that I've been conducting on our property in 2019. We're going to start with a quick intro about what RARE is, some things about our history for those who aren't familiar with the reserve, some of the things that we do, and then we'll get into the project itself. So we were founded in 2001. The Rare Charitable Research Reserve is a community-driven urban land trust, nature reserve, and environmental institute. Our headquarters and main property are located in Waterloo region and Wellington County, and we're on the west end of the city of Cambridge. Our property protects almost 1,000 acres of highly sensitive lands. The reserve is located within the Haldimand Tract, which spans 6 miles on either side of the Grand River from source to mouth, land granted to the Indigenous peoples in 1784 to recognize their support for the British in the American Revolution. For all of its properties, RARE acknowledges and is grateful to all of the original stewards of the land. Understanding that this land has been rich in diverse Indigenous presence since time immemorial, we'd like to honor and respect the sovereignty of both First Nations in our area, the Haudenosaunee peoples of 6 Nations of the Grand River, and the Anishinaabe peoples of the Mississaugas of the Credit. Miigwech to these nations who share their land with us and allow us to do all this great work. We'd also like to acknowledge the neutral people for whom we have archaeological evidence dating back 10,500 years. So coming into a bit more recent history, our slit barn and companion farmhouse, where many of our educational programs happen, is an important reminder that Mennonite families settled the area beginning in about 1800. In 1853, William Ashton purchased about 230 acres of this land along Blair Road, Cambridge. He's said to have named his property Crookston Park in honor of the Crookston Castle, the ship that brought him from England. Within a few years, Ashton sold the property to Matthew Wilkes in 1858. This property stayed in the family for a very long time. Eventually, Matthew Wilkes' grandson, Matthew Wilkes Kiefer, gifted the estate to the University of Guelph, which took possession of it upon his death in 1973. Kiefer's vision stressed the importance and need in the future to undertake research close to urbanization within a controlled environment, which is something that RARE has really taken to heart. So in December 2001, the Crookston Charitable Research Reserve was incorporated as a charity, and in 2004, we renamed it RARE. Its bold new name helped to end the confusion between this property and the private 53-acre Crookston Park and Crookston Manor, which is still in the area. So within the watershed, RARE is comprised of 24 different broad habitat types, including remnant old-growth forests situated in the Carolinian zone, where the Carolinian zone meets the northern hardwood zone. So we have all kinds of cool indicator species from the south and the north being in that wonderful meeting place. We have trees over 250 years old, and we manage the floodplain on both sides of the Grand River, where it straddles the Speed River coming from Guelph. As a key waterfowl habitat and critical flood regulation area, we consider this one of RARE's most important ecological features. Needless to say, we consider it highly important to steward these lands with respect for generations past, present, and yet to come. Since inception, RARE's taken care of the land on behalf of the community through the use of our three main pillars: conservation, research, and education. And I'm going to go into each of these just a little bit. Conservation is one of RARE's absolute top priorities. So through sustainable management, our lands and research sites are protected while the public can enjoy our trails, gardens, interpretive events. As the conservation technician, many of my responsibilities in this department include trail monitoring and maintenance, native tree planting, ecosystem restoration and habitat construction, and invasive species management. I run volunteer days throughout the summer focusing on all of these activities. And I also run our turtle conservation project, which protects nesting turtles and their eggs across Waterloo region in Wellington County. RARE also promotes the lands as a living laboratory for research, including in-house monitoring programs and partnerships with other institutions, community-based scientists, artists, and Indigenous peoples. Research informs our restoration practices and our education programs. We're a collaborative, multidisciplinary site that promotes research in all kinds of different perspectives: social sciences, humanities, psychology, biological sciences, and the arts, to name a few. We bring together experts, aspiring early career researchers, and the larger community. And we also offer scholarships of up to $4,000 to financially assist students tackling important environmental issues at RARE. We're actually currently accepting applications until April 1st, and I've included a link below for anybody that's interested. The Education Department at RARE provides a space for scientific research and Indigenous knowledge sharing for the children of our communities. More than 18,000 students have been through our youth environmental education programs called Every Child Outdoors, or ECO, which delivers K-12 programs that allow students to mirror research and monitoring activities conducted on the reserve. Our ECO camp runs weekdays through the summer and March break as well. We're also proud to have the largest community-owned community garden in Waterloo region, with 110 plots, as well as extensive education, demonstration, and food bank gardens. Part of the ECO program involves teaching the camp participants how to garden, and they get to eat a salad from produce that they get to harvest themselves. Every year, volunteers harvest thousands of pounds of produce for the Cambridge Self-Help Food Bank from our food bank gardens. In 2019, 5,000 pounds of produce was donated, and we recently added an orchard to our gardens, allowing us to now donate fresh fruit from our trees. Okay, so let's get into the project a little bit. So here in the Conservation Department, we do a lot of invasive species management at RARE. We value an integrated pest management approach, and we do our best to use invasive species management as a research opportunity, which is where this project began. So the Region of Waterloo Community Environmental Fund, or ROSEF for short, was established in 2011 to provide financial support to projects that enhance and restore the natural environment in Waterloo region. We received this grant in 2019 to conduct research on Phragmites control on our property, where it's been creeping into our Blair Flats floodplain area, which, as I said, is one of our highest priority areas to protect, as well as our Springbank farm, the location of our gardens. So first we had to design an experiment. To do this, we chose to compare 4 different Phragmites control methods. So spading, the first one, has been our default control method on the property so far. Hundreds of hours have been dedicated to spading Phragmites on the property, and it has had an effect, but it's incredibly time-consuming and requires a lot of volunteer labor for large-scale results, and we find that the Phragmites is actually outpacing us in some ways when we're only using spading as a control method, which was part of the reason behind this study. We had tried infrared treatment on the bottom left there, which is essentially mowing down Phragmites, clearing it from the ground, and then applying a heating element on the patch to bake it, basically, to bake the rhizomes. We tried that once on a patch in 2018, so this was actually our second year of treatment for that one. And then goat grazing and herbicide application had never been tried on the property before. So the first phase of our project was detailed mapping of all Phragmites patches on the property. We recorded information such as patch area, distance from streams and wetlands, etc. And we divided them into 3 distinct populations. We have our Blair Flats population, which was in the riparian area, the Springbank farm population, and then we separated out the east of Crookston Creek population just because there's that riparian area between and they are distinct. We gave every single patch its own unique name, such as BF Frag 1, like Blair Flats Frag patch number 1, so that we could easily communicate about them in the future. And we're hoping that these names will stick over the next number of years. In total, we mapped over 19,000 square meters of Phragmites on the rare property, no small feat. Of the total 20 Phragmites patches, we chose 5 for use in this project. So 1 was designated as a control, and the other 4 were used for each experimental treatment. So I bolded the plots. You can see here they were the ones that were chosen. And this was based on the level of priority in terms of how important it is for us to remove them from a particular habitat, the ease of access for equipment or goats, and the distance from provincially significant wetlands in the case of the pesticides. The majority of these were done at Springbank Farm, which we largely consider a staging area for our invasive species research and education. The only exception to that is our Blair Flats Phrag 4, which was used for the infrared treatment, just because we had to be able to get the equipment from Blair Road into a Phragmites patch. So in all 5 chosen patches, 3 plots of 3 meters by 3 meters were created. Information collected in these plots included everything from the UTM coordinates of the center, habitat type, growth stage, stand density, which was measured in stems per meter squared, all kinds of stuff. And this baseline data will be used in the summer of 2020 when we return to determine the effects of the treatment. We'll basically recreate the plots in the same area and measure all these same factors. So of course, the next phase was treatment. The first one was spading. Spading was originally taught to our staff by Lynn Short from Humber College, and we used these handy spading postcards created by the Ontario Phragmites Working Group to train new staff and volunteers on spading protocols. So for anybody that's not familiar, spading of Phragmites is essentially taking a sharpened spade, placing it at 45-degree angle at the base of the plant, and using your leg muscles to push the spade into the ground so that you can cut the stem below the sediment. It leaves the soil undisturbed. You basically pull out the stalk and tamp the soil back down to remove it. So the spading treatment took place at the beginning of August. It was aided by the crew of Leon's Furniture. It took 11 of us all day to get through the 3 experimental plots, basically from 9 to 5. So it was a very large effort, but we did make it through. For grazing, first we needed to find somebody who was willing to rent us goats, which actually took a lot longer than we were anticipating. Finding someone to rent you goats to eat invasive species, it hasn't caught on quite as much in Ontario as it has. It's a lot more experimental in the States. It hasn't caught on as much here in Canada. So after a lot of research, a lot of people who were willing to sell us goats but not rent us goats, we finally found Sarah Mills from Growing Hope Farm in Cambridge, and she was willing to rent us 10 goats after we'd purchased an appropriate enclosure for them. Goats have a very good ability to escape a great many enclosures, and she was worried about goats escaping and also wildlife on the property. It is a natural area. So she suggested something called the Cackalack 1312. A cackalack, or a chicken tractor as it's sometimes called, is a fully enclosed covered fencing structure with modular wheels on all 4 corners. The structure can be cranked up onto the wheels and then moved to a new patch of Phragmites once the previous one is sufficiently grazed. It came to us much like a piece of IKEA furniture in very small pieces with the instructions. We had a group from Fix, who work in IT, come to volunteer, and they helped us put it together. They did a fantastic job. So we got that thing together, and then she was willing to rent us the goats for about $100 per week because she knew that they would be safe, and it's very little work for the goat farmer if you're taking care of them and you know that they're safe and you're feeding them and everything. So we hosted the goats in our Springbank Farm Phragmites patch for a period of about 10 days, during which they happily grazed 1,040 square meters of Phragmites. Human labor was required, but not nearly as much as spading. We had to check on the goats once per day, top up their water if it was getting low, and then every other day we were moving the goat enclosure, which required about 5 people for an hour to roll it to a new site. Now we were moving the enclosure every day, or every other day, but you probably could get away with moving it every day. They ate it so incredibly fast. We were really impressed with how voracious the goats' appetites were and how much they really seemed to like Phragmites. So for the infrared treatment, a large swath of the grass needed to be mowed down both within the treatment area and around it. This was largely due to the fire hazard around the heating equipment. So we mowed down a very large area of Phragmites, and then the treatment itself took approximately 2 hours at the end of September. And there was the operator who was hired, and then 2 employees, including myself, supervising. So after it had been mowed, first the leftover vegetation was cleared right to the soil by the bobcat, and then a 300,000 BTU heating element was applied to the ground in an attempt to essentially bake the rhizome system. For reference, a home stove element on max is about 7,000 BTU, so this is quite a bit more powerful than that. As far as I know, this hasn't been tried before, so this is our first attempt at trying to bake the rhizomes without simply doing a burn. This is something a little bit different, so yeah. Finally, we did glyphosate spraying, followed up with a mowing treatment 2 weeks later. This is a very standard Phragmites control method. Spraying took place by me on October 8th, and it was mowed down in mid to late October. So the fourth and final phase of the project is still ongoing. It's site monitoring. It's difficult at this point to report conclusive findings because we simply haven't come back the following season to do that monitoring yet. But I'm going to briefly go through the results that we have so far and discuss what we've learned. First, we're going to look at the visual results, and then I've put together a fairly complex table that we'll go through kind of line by line. So as you can see here, 8 weeks after spading treatment, new Phragmites stems were emerging. There was regrowth from other species as well, but it was a lot thinner than it was before. It's possible that this regrowth was repressed. There were still quite a few dead stems littering the ground that we didn't remove. So that could be part of it, repressing both Phragmites regrowth and the native plants. The goat situation was very promising. This was the only treatment in which the removed stems were completely consumed, leaving no dead stems or piles behind, which might explain the greenery in the picture. Phragmites was observed returning to the site, but we were surprised at the lack of density in the Phragmites and the density and diversity of the non-Phragmites vegetation that was coming back 10 weeks later. This could have something to do with the fertilization of the soil while the goats are eating the Phragmites as well. So I'm really curious coming back in 2020 to see what exactly this is going to look like in a following season, especially if we come back into another set of grazing. So infrared was certainly effective at killing the above ground vegetation. The soil was still completely bare 5 weeks after treatment. This was at the end of October. It kind of looks like the surface of the moon. With the lateness in season, it's not totally surprising. However, as this is the second year of testing, we do have some results for infrared between 2018 and 2019. So as you can see, we do have some indication of infrared having an effect between 2018's treatment and 2019 before we mowed all that frag down. You can see that the height of the grass is slightly less on the treated side of the patch. And when we investigated further, we did find that the treated side also had more non-Phragmites vegetation within the stand. On the right side, we did surveys and we couldn't find a single thing that wasn't Phragmites other than a couple really unhealthy looking elm trees poking up over top, which you can kind of see in the picture. When we were in the side that had received the infrared treatment in 2018, we did find other plants within the stand. So that was promising. I'm looking forward to seeing what the second year of treatment will have done. And as you can see, similar to the infrared treatment, it's difficult for us to evaluate regrowth so late in the season, 5 weeks after spraying and 3 weeks after mowing. There were still only dead stems. Again, this could be because it was so late in the season. It was because of the amount of work that had to be done. It was necessary for us to stage these different treatments throughout the season. So this was the final one, and this was getting into November. So it's difficult to say what the results are going to look like until the following growing season. Okay, time for the big table. So with the information we were able to collect this early in the monitoring phase, we did our best to create a table of the known data that we had gathered so far. Spading was easily the easiest control method to acquire. It was just having sharpened spades. And we could also access both terrestrial and aquatic sites, the easiest with spading. Although, with the exception of aquatic areas, the backpack sprayer was quite easy to get into sites with as well. It's just that, of course, it doesn't matter if you can get to aquatic sites with a backpack sprayer because you can't spray in them. So there's that. Grazing was more difficult just because of the enclosure that we had. It was only possible to get to where we were able to roll the goat enclosure to. And that made it really difficult for us to access a lot of sites. Hopefully this year we're looking to experiment with hooking the enclosure up to the back of our tractor and maybe dragging it somewhere. But we could also experiment with different kinds of fencing. However, if you're looking at portable fencing, it's a lot more likely that goats will be able to escape. So we'd need a lot more supervision and staff or volunteer time dedicated to making sure that the goats are safe. The infrared requires site accessibility from the road, or at least that the bobcat machine can get to the site. This worked for quite a few patches along Blair Road, but certainly not all of them, and certainly not our aquatic sites. In terms of affordability, spading was the most affordable treatment. We already had the spades and the volunteers were free, so it actually didn't cost us anything other than time. Though the rolling goat enclosure, the cackalack, was expensive, it's approximately $3,000. Once that investment has been made, you're really bringing your long-term costs down because the goat grazing services were quite affordable at about $100 a week because there's so little time on the farmer's part and on the staff's part that's required to ensure that the goats are safe and that the Phragmites is being eaten. Infrared was a little bit more expensive. For the 2-hour treatment, getting the equipment out to the site and everything, it was around $2,000. So a little more expensive in terms of the amount that you get. And then herbicides are expensive per jug, but ultimately I was really surprised at how little we needed and how high the dilution factor was. It's ultimately very cheap per square meter treated with herbicide. Now, of course, all of the treatments are organic except for the herbicide, which is especially important when we're looking at the areas adjacent to our community gardens, for example. If there's people that want to ensure that their gardens are staying organic, we can't be spraying herbicides immediately adjacent to the community gardens, etc. And then damage to the rhizomes is a really key factor for Phragmites control. Simply mowing them above ground has been shown to have little effect or even stimulate growth. Phragmites are very much rhizome spreaders. A lot of their growth, a lot of their vigor is contained underground. So it's important to consider how you can damage the area underground. I like to think of it kind of like a hydra. So spading and herbicide have already been shown to be effective in this way. By cutting the stems underneath the ground, you're making it a lot more work, a lot more energy for the rhizomes to send up more shoots, which can ultimately degrade the amount of energy that they have stored in them. Herbicide, of course, you're translocating the glyphosate down into the rhizomes. Grazing doesn't seem to damage the rhizomes. It only damages the above ground vegetation, which means that repeated treatments or perhaps alternating treatments may be necessary. We've been looking at potentially going in and grazing an area to remove all of the above ground vegetation, and then for repeated treatments, looking at doing spading after that. And then theoretically, infrared baking of the ground should damage the rhizomes as well, but of course, because it hasn't been tried before, we don't have absolutely conclusive proof of that quite yet. The only treatment that consumed the dead stems themselves was the goat grazing. We found that incredibly helpful, and it really seemed to help for the regrowth as well. So as you can see, the vast majority of the check marks are on spading. it's easy to acquire, it's cheap, accessible, all this stuff. However, there is a very key negative with spading, which is the labor requirement. You can see it's more than 3 times the number of person hours required than any of the other treatments, and that's because you're focusing on it one stem at a time. And when you're looking at like a 3 meter, not even like a 1 meter by 1 meter plot, you're looking at densities of up to 200 stems per meter squared that you're not going to get anywhere just doing spading. So that was something really important that we were looking at specifically, is that labor requirement. So our last 3 rows here focus on the effects of the treatment so far on both the species of Phragmites and the other stuff around it. So spading in theory should have low vegetation and soil disturbance. But realistically, when using volunteers, we didn't see the level of precision and accuracy that might be required. We found a lot of issues with the volunteer spading above the ground, or a lot more soil disturbance than was completely necessary, that kind of thing. However, if you have the same volunteers over and over again, as they become more skilled, the disturbance does go down of soil and non-target vegetation. The regrowth of native plants started after 8 weeks, but it was fairly thin compared to the Phragmites that was coming back. Grazing had a very high level of vegetation destruction. The goats actually ate every single thing within the enclosure except for the dead teasel stems. They ate the thistle, they ate the Phragmites, they ate everything. However, the soil disturbance was very low, and it might even actually have had a net positive effect, considering the fact that they're fertilizing that soil as they're eating. And after 10 weeks, a thick layer of non-Phragmites vegetation was growing back. So that's really promising for the grazing treatment. Now, infrared treatment, of course, had the highest vegetation and soil destruction by far. The treated site was reduced to bare baked soil, and after 5 weeks, no regrowth was seen on the site, though it may have been due to the late season. It also could have something to do with the microbiome of the soil, like the microbiota within the soil may have been baked along with the Phragmites rhizomes. So it's possible that when we're considering a restoration plan after the infrared treatment, looking into soil amendments, because that could have something to do with it as well. However, it should be added that we did observe a higher density of native vegetation versus Phragmites between 2018 and 19. It's going to be very interesting to see what it looks like after 2 years of treatment. Finally, the herbicide treatment likely involves some vegetation destruction due to unavoidable glyphosate drift, and also plants that are within the plots where you're trying to spray. It's quite difficult to avoid getting a little bit on the plants that aren't Phragmites, especially if you're in an incredibly dense stand like we were. But soil disturbance is fairly low. No regrowth was seen between the October treatment and the end of the field season, but again, the treatment was done in October, so there may not have been time for things to come back. Overall, I think it's safe to say that choosing an appropriate Phragmites control method is complicated. It's dependent on the site, the money, the labor, the time available, what your specific goals are, how many patches you have on the property. There's so many considerations, but I hope that this is helpful in terms of deciding which treatments you want to look into and what could be promising, what could be paired well together if you want to do 1 year of, say, grazing, follow it up with a spading treatment, that sort of thing. It's nice to have some baseline data. So moving into 2020, we're going to be looking forward to seeing what results we can collect over the next year. So we're going to be going back to all of the same 5 patches. Every experimental plot we're going to be recreating. We're going to be evaluating change in all of those variables we talked about before. We're going to be looking at the changes in stem density, the diversity of non-Phragmites vegetation, etc. Hopefully, we'll be able to find sufficient funding to continue the experimental treatments in this coming year as well. Of course, spading is fairly easy, but we're of course going to have to find funding to bring our goats back. We're going to be hopefully able to afford the infrared treatment again, although we're not sure about that one. So hopefully we'll be able to find funding to continue these projects. And just wanted to talk a little bit of a side here. My other main invasive plant project that kind of ties into this work that we've been doing is the updating of RARE's invasive species management plan for 2020 to 2025. We're largely basing a lot of our work on the Credit Valley Conservation's amazing work that they've been doing with invasive species management and plans and the stuff that they've been putting out, as well as the Ontario Invasive Plant Council and their best management practices. We're looking at a very holistic view of the property. It's a very large property to come up with a management plan for. So we're going to be creating tailored restoration plans for different specific areas of the property. We're going to be looking at invasive species management. What are our priorities for this old growth forest, and how is that different from our invasive species management that's taking place in that riparian area? So we're going to be having mini invasive species management plans taking place in all different areas of the property. We're going to be looking at an integrated pest management approach, looking at all different ways that we can control invasive species, and take opportunities for research whenever possible. As I had said, we're still accepting applications for our scholarship programs through RARE. So if people are interested in linking those things, it might be something to look into. If anyone's interested in talking more about this plan later, I'd be happy to discuss it, and definitely keep an eye out for when it finally gets published. And finally, I wanted to do a big thank you and shout out to the many organizations who've helped with this project, and very much to the Great Lakes Phragmites Collaborative for having me. Shout out to the region of Waterloo for funding much of this work, to Growing Hope Farms for bringing us those amazing goats. Big shout out for them. They also do things like birthday parties, they do goat yoga, they provide goats for weddings. It's a very cool urban farm in Cambridge. Thank you to Fix for helping us build our goat enclosure, to Leon's for helping us spade all of that Phragmites, to Heat Design Equipment Inc. for providing us with that infrared equipment, the Ontario Invasive Plant Council for inviting me to talk at their conference, which is where I met Sam. And thank you for having me to do this talk. It's been very fun, and I look forward to seeing what questions are out there. So yeah, thank you.
Thanks so much, Sarah. That was wonderful. And I think I speak for everyone listening in that if you organized a goat yoga, we would do that.
We'll have to look into that. We actually do yoga, like we do a yoga in the gardens in the summer already. So maybe there's something there that we could do.
That's great for us. Well, yeah, we definitely have some questions. I'd like to apologize to everyone out there. It looks like GoToMeeting broke their own link shortly before the webinar started. So I'm sorry to everyone who joined late. This webinar is being recorded, however, and so if you missed the first few minutes of it, I'll send out that link later today. And I would definitely go back and give it a listen. So yes, we're taking questions. You can go ahead and type them in that chat box there. And if you need to leave the webinar early, that's no problem. Like I said, it will be recorded, and we'll send that link out as soon as it is available.
Okay.
And we'll just move on to questions. And if you have to leave early, this is Sarah's contact information as well as our contact information below. So the first question that we had, Sarah, people had a few follow-up questions about your herbicide treatment. Can you confirm which herbicide was used and what the dilution was?
Oh, yeah, for sure. So we used Roundup, Weather Max, glyphosate herbicide, and the concentration of it. We based it on the label on the Weather Max for Phragmites. It might have been 5%. I would have to go back and look through.
Okay. If anyone has any questions, I'll just encourage you to follow up with Sarah, but that's really helpful. Just to follow up on that one, we did have another question from Heather, and Heather wants to know if you followed the BMPs for herbicides, which includes the removal of standing dead vegetation prior to the application.
So the standing dead vegetation, we didn't have a huge amount of it in the patch that we chose to treat. We did have to choose separate patches for each of our experimental treatments just because of how small the patches were and like how many plots that we could fit in them. So the one that we happened to choose for the herbicide treatment didn't have a very significant amount of dead standing vegetation, and we didn't actually remove the dead vegetation beforehand. We just mowed it all afterwards.
Great. And then we have a question from Tim. Tim wants to know what kind of treatment was used after spading. Some groups cover the ground with tarps or do tarping to prevent reemergence. And did you have any sort of replacement planting going on after the spading effort?
That's a good question. We haven't planted anything yet. We are currently looking into restoration plans for post-Phragmites management. We are anticipating that we're going to have to respade the area, so we haven't done any plantings yet. But we're actually looking into a lot of research that's been done by Niagara College, actually, where I graduated. There's been a lot of work done on dense live staking of willows for preventing the regrowth of Phragmites after treatment. So that's something that we're planning on doing in the areas where we've spaded. It hasn't been done yet, but it's included in our invasive species management plan, and we're hoping to go ahead with that in 2020.
Great. And just a clarification, sorry, Heather, I asked her question wrong. You did answer it already, Sarah, but she was referring to removing dead vegetation after treatment.
Yes. So yeah, we mowed it all down afterwards.
We do have another question about spading. So this is from Emily, and Emily wants to know, she really is a comment, this is going to be a new technique to me. Is the goal to cut stems low enough that they're getting flooded? And she's concerned that this isn't really addressing the rhizome. So can you speak to what spading is really doing?
Yeah, so there's kind of two strategies there. Spading can be done if you're looking at an area that does get regularly flooded. There has been research showing that cutting the Phragmites below where the flooding happens can effectively drown it in a way. However, in some areas, it has been tested in dry areas as well, which we also did. We did an aquatic site outside of this project, and we also did a dry site. And a lot of it is also cutting it below the area where it's easily able to send up the shoot, so it has to redevelop that tissue. And a lot of it is taking the energy out of the rhizome. So it's not the sort of thing that you could look at having one treatment. But if you have repeated spading treatments, typically twice a season, over a number of years, you're going to be seeing the energy inside the rhizomes decreasing so that they're not able to put up as much of a fight, I suppose. I think that it's a great thing to have along with other management techniques. But yeah, that's essentially, yeah.
And I will let everyone know that if you visit the website that's linked on the screen right now, GreatLakesPhragmites.net, we do have best management practices for spading and herbicide treatments up there as well. And we do have the postcard image that Sarah had showed earlier in her presentation on spading.
Yes.
Heather has another question. She asks, based on your work, what size of management area would you recommend for using goats on?
Oh, yeah, that's a really interesting question because, of course, we did it on a very small scale. The original study that we based our goat treatment on was a much larger Phragmites patch. They had a much bigger budget. This was a university in the States that essentially had like a giant patch of Phragmites, and they had their goats on it for a much longer period of time. We were looking at, I think it was something like a 40 meter squared area for these plots. So I think that grazing can be used on many different sizes of Phragmites patches. What you're looking at in that case is what your stocking density is like, and that just goes back to rangeland management theory, where you're looking at, okay, what is a amount of grazing that's going to take out this Phragmites? Okay, how many goats do I want on this area? So I think grazing is really great because you can tailor it to a really small site or a very large site, just depending on how many goats you have available and what kind of fencing you're using and that kind of thing. So it's definitely dependent, but I think that it's very manageable that way.
Great. And then we do have another question about coordinating volunteers specifically for spading. We know that you did show that the amount of effort is required for spading is quite a bit higher. I think you said 3 times higher.
Yes.
How was that compounded by trying to organize all of those volunteers and coordinate those efforts?
Absolutely, yeah, it is difficult. I think that if you had something like a spading task force that like was very well trained, you could almost contract it out if it became like a really mainstream control method. But I think that a huge amount of it had to do with having volunteers that are typically untrained. But that's also, I think, realistic for a lot of environmental organizations. Many of our volunteer days, we have one-time volunteers coming out, or we have high school students coming out trying to fulfill their volunteer hours that they need for a particular course. And then we don't see them again. So you don't have like trained professional Phragmites spaders coming out a lot. I think in the environmental field, it's these volunteers from the community who are very well-meaning, but you need to put that time in to train them. And that is an investment for sure. If this became more mainstream, I'd like to see that number go down. I think absolutely it's possible.
Great. We'll just wait one more second. If anyone has any last-minute questions, you can go ahead and type them into the chat box now. And sure. I'll say once again, the webinar is going to be recorded. And so if you would like to go back and rewatch or listen to the beginning or share out with other partners who may be interested, please do so. I'll send out that link later today, and it will be available on the GreatLakesPhragmites.net.
Mm-hmm.
Okay, well, it looks like those are the questions, so I'll just go ahead and reiterate this is going to be available. Thank you so much, Sarah, for your great presentation and for sharing your work with us. I knew there was going to be a lot of interest with the different treatment options that we don't normally hear about, the infrared in goats in particular. So thanks so much for sharing that with us today.
Absolutely. And please, to everyone, feel free to come by Rare Charitable Research Reserve in the spring and the summer. We have 8 kilometers of trails that are free and open to the public. I highly recommend going for a hike. We've got everything from limestone cliffs and alvars to old growth forests. It's a really great area to visit for anybody that's interested in ecology, for sure.
Absolutely. Thanks so much. And as we wrap up, I'd just like to acknowledge the U.S. Geological Survey and the Great Lakes Restoration Initiative for providing the support to the collaborative. If you have any additional follow-up questions, please reach out to us at phragmites@glc.org. So thanks so much, everyone, and have a great day.